It's my great pleasure to introduce our speaker for today. Jaromír Gumulec is an associate professor at the Faculty of Medicine at the Masaryk University in Brno. He has both a doctorate in medicine as well as a doctorate in human physiology and pathophysiology from Masaryk University. And he is part of the cancer research lab led by Michael Masaryk. And his research and area focuses on cancer mechanobiology, linking and bridging biology, mathematics, and informatics to focus on biomarker discovery and cancer resistance. And we look forward to the presentation, Jaromír. Please take it away.
So it is my warm pleasure to receive such nice introduction. I'm glad to be here today with all of you and to share information, share my slides about microscopic technique I'm using. So as Johanna nicely said, my background is a cancer research. With this I focus on topics of how cancer cells are becoming aggressive ones. So apart from studying relatively routine cell biology assays, we are using quite effectively approaches like atomic force microscopy to understand the biomechanics of cancer cells or the extracellular matrix. And I'm as by education I'm a doctor of medicine. I'm always I keep always questions about how the findings could be translated to a clinical setting. What is the potential of a biomarker or some diagnostic tool of such imaging or such approach. And also with this I approach cancer cell mechanobiology. So let me start my talk. Last decades, 80s, 90s, were about a tremendous amount of research done on molecular biology, linking cell fate with a spectrum of molecular regulators. However, what we see now more and more is that it's not just about molecular biology. The cell fate is dictated also about morphology of cells and the cell mechanics. Given that, well, it is important to have a look on the cell mechanics from the perspective of cancer biology. Well, it's simply because the mechanical stress which is applied to the cells is kind of a rate limiting step for cancer progression. Cancer cells simply need to be adapted to the extracellular environment, the microenvironment, physical properties. So they have to have strong enough actin cytoskeleton in order to survive. But in the same time have to be deformable enough to enable migration through confined areas. And well, with that, among a spectrum of techniques which is usually used in field, a number of results are reporting that there are cancers which indeed, compared to a non-tumor counterparts, are either being stiffer, or in the same way there are some others which are being softer compared to a non-tumor counterparts. So it's rather about being directly stiffer or softer, about finding kind of optimum of cell stiffness with regard to the environment for optimal invasiveness and migration of cancer cells. And well, to study this, the most common approach is to do with atomic force microscopy, which for like live cell imaging has become like a standard, and indeed is also used by our lab very frequently. But as we were using this technique, we perceived that, well, we in some experimental setup are limited by relatively slow acquisition and also relatively unphysiological mechanical stress, which happens throughout the cantilever and the sphere compression of the system to the cell. So we were asking, okay, so is there any approach we can maybe modify or develop or employ in the lab, which may be more high throughput, or which may be more physiological for the cells? And what came on our minds is that, well, what is also a physiological rate limiting step for cancer cells is a shear stress in the circulation, because the blood is one of the most typical channels through which cancer cells invade to form a metastasis. So therefore, we decided to like simulate a shear stress in the chamber as a stress to induce cell deformation. And intentionally, we used shear stress levels in our experiment with quantitative phase imaging, which normally happens in arteries or relatively benign lesions of the human circulation. So to do so, to simulate the shear stress, which happens also to cancer cells, we recapitulated this setup by using a system with a pump and a flow meter, so we can stress the cells by the flow while continuously measuring the differences in the position of the cells. And because we have the flow meter information, we can also get the information about the actual shear stress levels. To do so, we are using various chambers based on the shear stress necessary. So for a traditional setup, we use flow chambers from EBD, which are 100 microns high and 1 millimeter wide, to which we seed cells, and then you can very easily model what kind of stress is then applied for the cells, what is based on the flow, what in particular part of the chamber where the cells is, what is the actual shear stress in every single cell in the sample. So the first question here is to identify the stress, which can be quite easily calculated based on the geometry and the flow parameters. And then the second question comes to define a strain of the cells. Well, and to define the strain of the cells, imagine a cell sitting on a surface on a 2D culture, which is deformed by a flow, by a pulse of the flow. So as this happens, the center of mass, this COM, moves in the direction of this flow, and when the flow goes off, then the cell returns partly to the original position. So by measuring this angle of deformation of the cells, we can have a proxy to elasticity of the cells. Well, to know this angle, however, we need to know the height of the cells. And here comes the question. So how we can measure the height of the cell? What is the most common approach? The easiest one to do in every such a lab is, well, to go confocal, to do confocal slices. And so that you stain your cells and you can easily measure the height of your elements. But well, as you are doing confocal using a routine confocal setup, it takes some time. You need to slice the whole sample to get this information. And you also need to stain the cells with some fluorescent marker, which may alter the properties of the cell. So how actually to solve this? And there is a way to have a look on this topic, because you don't necessarily need to rely on the intensity property of the light, but you can also have a look on the phase property of the light. As the light passes through a sample like a cell is, well, the wave front which passes through a sample is being distorted, and this distortion of a phase can be detected. So why it is good? Why it is relevant for identification of height of the cells? Well, it is the phase shift you actually are measuring, and the phase shift is then proportional to the cell height under given refractive indices of the sample. And well, to measure this, we are using a quantitative phase imaging, and specifically coherent controlled holographic microscopy, which enables to measure the phase shift simply by a technique in which the sample, we don't just measure information on a sample, but also through reference sample, and reference sample is a sample which differs just with the cells. Otherwise, it is optically identical. And as those two beams are interfering on the CCD camera, a hologram is being created, and from the hologram you can mathematically easily calculate the quantitative phase image in which comes as a phase shift in a radians. And this you can then quite easily mathematically recalculate after with some limitations to cell dry mass distribution, for instance. To do so, in the last decade, several techniques appeared out commercially, like holomonitor system, nanolife systems, TomoCube, or the T-Light solution, which is based in Czech Republic. So in fact, it's relatively not necessary which quantitative technique you will use for such approach. If it fits your sample, you are pretty fine. So we are using the system from T-Light, the Q-phase microscopy, but it's actually, if you measure phase shift, it's not relevant which microscopy exactly you are using. So in fact, if you are measuring phase shift, you are actually measuring physical property. So you don't need to use any fluorescent tags or fluorescent labelings, which, as we perceive and we think is or could be a great advantage. You are practically free of phototoxicity. You don't have problems with photo bleaching, and we are using light of 650 nanometers, which is a relatively little harmful to cells. So we, using this technique, can also do quite long measurements. This is actually not necessary for my assay, but this is possible to be done. So we are label free, and because the technique is quantitative, it means that from phase shift we can also approximate cellular parameters like cell dry mass or cell dry mass density of the given sample. So you are in fact not measuring the intensity of pixels, but physical parameters of the sample, which do not change of that particular solution. So this is also an important finding of this microscopy. So quantitative phase microscopy, label-free technique. The first, what we need to do is if we want to measure the physical properties or the elasticity and viscosity of individual cells, well, we need to identify individual cells in a sample. So, well, phase contrast, which is one of the simplest label-free technique, is, well, actually also label-free bright field technique. So, can I use also this, or can I use DIC? And where is the difference? Why it is for us meaningful to use quantitative phase imaging? Well, the advantage is, as I said, in the quantitative character of the image you acquire. You don't get like halo artifacts which are introduced in phase contrast, or these shadow-like artifacts like in DIC. And mostly the most significant benefit is that you are actually measuring data, not some relative intensities of pixels. And this is not only beneficial for data analysis, but also for the first necessary step you need to do. And this is the image segmentation, because QPI data is far easier to segment than any other data. But we got it into trouble. Okay, so we need to analyze QPI data. How to do so? So which algorithm of cell segmentation we could employ? So back in 2018, with Tomáš Vičhar and Florian Juk and other colleagues, we asked exactly this question. So what would be the optimal solution for such imaging technique? So we've performed kind of selection of optimal segmentation technique, and based on this, finally we can employ relatively simple detection and segmentation approaches based on distance transform and thresholding. This enables us relatively satisfying results. However, as time goes by, the deep learning also was important for our image analysis, and back in 2021 we also introduced UNet for refinement of cell segmentation and tracking for the analysis of quantitative phase image. Because, well, if you track the cells, you need to have them tracked precisely throughout your measurement. Otherwise, you can easily get very altered results. So once we have our segmentation done, we can step further and to really perform the experiment. So we are exposing cells to a shear flow pulses, which we gradually turn off and turn on and turn off. To control this, we built a really cheap solution with Arduino-based pump controller and a simple pump from even some 3D printed components and some hobby market components. And this we connect to a chamber with cells followed by a flow meter. And this enables us with EBD chambers, which we use most often for this approach, we are capable of doing pulses of shear stress and to control them in a designated software we develop. So this is the approach of creating stress for the cell. And well, to receive the information about the strain, we record videos of which looks like this. What you actually see here is a phase shift time lapse, and the information is coming in radians as it is a phase shift. And what we are interested in is that when we typically stress the cell 3 times with 3 intermittent pulses, we are, once we have segmented cells, we are tracking the movement of center of mass of individual cells. So we identify the center of mass of individual frame, and based on how big the shift of the center of mass is, the bigger, the higher the elasticity is. So from such a cell, we could get a recording which resembles like this. What you have is a recording over time, so that you can see 3 pulses, and on the y-axis the shear strain. So the height of individual pulses are proxy for the elasticity. Then, well, the steepness of the individual pulses are then proxy for viscosity. Plus, well, you can to some extent also see that the cell shifts with individual pulses, which tells us a little about the adherence to the substrate. And well, given this, using the simplest Kelvin-Voigt model, you can calculate the viscosity of the measured cells. So this we usually visualize in a way like this. So we show the image of the same cell before the pulse and in the maximum deformation throughout the pulse. So we get the profile of this deformation and the deflection of the center of mass. So from this, you then can calculate the shear modulus as well as the viscosity of your cells. This you can do relatively fast based on how big your field of view is. You just have to wait like 30 seconds until you reach approximately the maximum of the deformation of your cells. Then you release the pulse. And well, because the assay is pretty fast for the acquisition, it can be quite well used also for analysis of relatively fast processes like actin polymerization, actin depolymerization. And apart from this, we observe that when we compare, so here we just are comparing cells treated with cytokalasin D to inhibit actin polymerization, and docetaxel to modify, to stabilize tubulins. We observe that there is a pretty good agreement with Young modulus determined by IAFM. So despite the shear modulus and Young modulus numbers are different, there is relatively good agreement in the trend between those two assays. And then for as I use cytokalasin, you can use cytokalasin to relatively dynamically polymerize and depolymerize or inhibit polymerization of actin. On a model of prostate cancer cells, which differ in actin quantity, we can use several interesting phenomena using this assay. So the 22RB1 cells, which are expressing relatively low amounts of actin cytoskeletal proteins, you can see that the cortex of those cells is relatively weak. So you can see that upon the pulses, you observe such a blebbing of the membrane, which, if you do not use excessive shear rates, are returning to the original position. And you can use also the cytokalasin in a time-lapse experiment, where you can measure the rate of repolymerization after a cytokalasin washout from the sample. So this you can measure like dozens of cells, and so throughout the time you can then see how the polymerization restores And then, according with this, the shear modulus restores to original pretreatment levels. In such a robustness, this cannot be that easily done with routine AFM setup. Now, as you stimulate the cells by the flow, you kind of expect that the pulse you act on a cell is of a square shape. This is just what you expect. This is in many scenarios, however, relatively difficult to maintain. There is the properties of the tubing we are using, property of the syringe, which puts a capacity to the system. So instead of a square expected ideal square pulse, you get something like this with a glass syringe and with the most rigorous tubing you can have. But in a relatively usual syringe and just normal fluidics, you rather get signal like this. Well, which is definitely a problem for measurement of for the accuracy of a viscosity determination, because for viscosity you really need the pulse to be as square as possible. But well, there is a hack how to overcome this limitation even with a flexible fluidics. Well, because in the system we are measuring the flow, and as we measure the flow, we then approach that the strain, the deformation of the cell is in fact a convolution of shear stress and the impulse response of so-called impulse response, as it would be a convolution. And the impulse response is actually the unknown parameters, which are nothing but cell viscosity and the shear modulus. So the fact that we know the actual flow in every minute or every second of the recording enables us to using the convolution to determine or to correct this for optimal flow. So this approach seem to be very effective in calculation of viscosity. So here you can see a comparison. So when uncorrected with just soft tubing, you have quite a high variability in the data, and when you have near square stress, so you should have lower levels of viscosity and also much homogeneous data. And with the convolution, you very well approach to this from the original soft tubing data. So this parametric deconvolution seemed to just be very effective, and based on this, we are employing the flow measurements routinely in the setup. Well, in some of the previous slides, I also mentioned that we use QPI because we need to have the information about cell height. And well, if we have a look again to this equation, so you have the cell face which you actually measure from the microscope. Then you have here you have the refractive index of the medium solution which you wash the cell. So you can easily detect this with a refractometer. And you have also here this the cell refractive index. So in order to determine height, you need to know the cell refractive index. So how to get the refractive index? And this is another approach how we use the quantitative phase imaging. Because you can modify what is outside the cell. You can modify the extracellular refractive index. There are various compounds. For us, what seemed to be very effective is a solution of yarixanol, which is of high refractive index. And using a setup with 2 syringes, you can intermittently change the extracellular solution with either just a cell medium and iodoxanol. So you get then recordings which looks like this. So you measure the phase shift of relatively high refractive index iodoxanol versus normal medium. So by such exchanges, you get finally 2 images, one with medium 1, one with a medium 2, which obviously differs in the phase shift you determine. And from this, you then can calculate 2 other images. One is the refractive index, and directly you can also calculate the actual height of the cell for given refractive index for every single pixel you have. And if you compare the refractive index versus height image, you can observe that while up in a phase shift image, you can see like a relatively low phase shift nucleus and high refractive index, high phase shift nucleoli. The same, even more pronounced, is evident in the refractive index image. This absolutely diminishes in a height image, where you can see very very gradual profile of the cell. This is like an important refinement. So just by adding another syringe, you can much accurately measure the height of your elements in a sample. Well, refractive index anyway is a interesting parameter for a cell biology, because it is an inherent physical property of the material, relatively constant to various materials, and for various materials, it changes in a way that with increasing concentration, you see an increase of a refractive index of the solution. And this steepness is described by a refractive index increment, a parameter characteristic for various materials. Generally, for most proteins and for DNA, it is approximately the same. However, for lipids, this number is quite different. So based on the cells you are working with, you should also consider to use this knowledge to just be reflected in the calculation of cell height. Otherwise, you can get distorted results. And well. What is also interesting with a refractive index itself, or with a light phase shift, is a finding that actually even the refractive index itself correlates with AFM-based, AFM-determined cell stiffness. So no matter of using the shear stress assay I demonstrated, also the phase shift directly provides some guidance about the physical properties, the biophysical properties, the stiffness of the cells, and of course it makes sense because, well, it reflects the amount of proteins, notably the actin cytoskeletal proteins, which are responsible for stiffness. And as I am slightly approaching to the end of my talk, I just want to highlight the what I perceive a big advantage of such approach. So, and this is that you can measure fast, relatively high amounts of cells. So we are measuring whole chamber with like thousands of cells within just 1 minute, which is quite a big improvement compared to like measuring with AFM. And you just don't get the information about the deformation, but you also have the routine information about morphology. You record from quantitative phase imaging. So you get just normal morphology parameters like size, diameter, sphericity, or whatever else you need. Plus, you get also parameters like refractive index, cell dry mass, and this together turns out to be very important when you reflect mechanical properties of the cells. Because, well, in relatively common AFM setup, when we measure, we just measure like 30 cells per well, then we go to like another treatment and measure another 30 cells. So it is feasible within one day to just acquire like a reasonable amount of data. Here you are measuring rather thousands of cells per experiment, and this gives you opportunity to see correlations between biophysical properties of cells and cell height, cell area, or mass density, or refractive index, or even with cell migration, if you also track migration for a short period of times. Which, as we perceive, turns out to be mutually very beneficial to know how these parameters cross interact and tell better or more complexly tells us the whole story about what biophysically happens in the cells. And well, when you have the cells growing in some clusters, you can see how the deformation differs in the front of the cluster, in back or inside the cluster, where is a much higher amount of cell-cell junctions. So this is also, I guess, an interesting finding you can get from this device. And well, with this, I'm in the very last slide. So we are using this setup not in order to just say goodbye to AFM, but rather in a way that we are combining the advantages of both systems, and especially for flow-induced cell deformation using quantitative phase imaging. We are using it because we are label-free. We are using physiological stressing, relatively physiological stressing to the cells. We can measure also the viscosity, plus biophysical parameters like cell dry mass, plus normal morphology. And because the system has just a fluorescent module, you can use this to with your probes, like for cell cycle, or whatever you need to just classify your cell according to your needs. And with this, I would just like to thank you for your attention, and also show you maybe the most important slides to show people without whom it would not be possible to realize this. I in particular really need to highlight the help of two first guys, Tomáš and Jan. Tomáš does the image analysis and hardware modifications and so mostly the bioinformatics and mathematics, while Jan is expert in quantitative phase imaging, and with those two and also others, I was able to develop this essay to a situation or to a condition which I can demonstrate to you right now. So thank you for your attention. It has been a pleasure to talk to you in this session.